A single nucleotide variation, or SNV, that changes an amino acid to a stop codon like this is called a nonsense mutation. Nonsense because it makes nonsense after this. There's nothing left. It stops translation. A single nucleotide variation, or SNV, that changes an amino acid to a different kind of amino acid is referred to as a missense mutation. It's also referred to as a non-synonymous mutation. And an SNV that doesn't change the amino acid sequence at all from the original sequence is called a silent or synonymous mutation. Now, silent mutations can accumulate because there's really no selection pressure on them, and they don't detrimentally affect the overall protein product. So one of the most common kinds of SNVs we find are silent mutation SNVs, where someone may code for a valine using a GTA, and someone else will code for valine using a GTG. Because the overall protein is unchanged, there's no direct selection pressure that can happen on the protein. It would be completely random which of these alleles would fluctuate in the gene pool. Now, missense mutations could be advantageous or detrimental. It depends entirely on which amino acid is replaced and what it's replaced with. Remember, structure is function, and in this case, it all depends on what the structure of the amino acid we're coming from and the structure of the amino acid we're going to. And then we have nonsense mutations, which are almost always detrimental because they're going to shorten a protein sequence. Once a nonsense mutation has been induced, everything past that mutation is not going to be translated. Take a look at this long sequence right here. I've given you two mutant versions. They both have nonsense mutations, a TGA here, a UGA in the genetic code, and a TAG here, or UAG it would be. Both are stop codons. I want you to tell me which of these two nonsense mutations is going to be more severe, and then I want you to tell me why in this text box right here.